I want to thank everyone for being here. I'm really pleased to be at Cornell. And I want to tell you about my research dealing with patterns of evolution in domesticated apple. So I'm really interested in domestication genetics broadly, and I have two major questions that I'm concerned with in my research. And one of these deals with broadly understanding how different evolutionary forces interact to either increase or reduce genetic diversity in domesticated lineages relative to their wild populations. And understanding this process involves understanding the interaction between two major types of evolutionary forces. And those forces fall into two main categories: stochastic or random forces, things like genetic drift, and deterministic forces, things like natural and artificial selection. And then, of course, like many people, I'm also quite interested in the genetic basis of phenotypic traits. And in domesticated species, I'm really interested in the origin of domestication traits, things that are useful in an agricultural environment. And I'm interested in the origin of those traits or those alleles in a temporal and geographic sense, and just understanding the mutations and the genes that underlie these traits. So today for my research, I'm mainly going to talk about this first question, understanding how these forces interact in domesticated lineages to affect genetic diversity in crops. And I work in plant crops, despite the fact that I have these lovely pictures of all different types of domesticated species down here. So let's talk about the interaction between different forces in domesticated lineages for a minute. I think most people in this room and in Geneva are comfortable with the idea of the role of selection during domestication, right? So in domesticated plants, we see to some extent a suite of characteristics that have evolved in many different species of domesticated plants. And these plants have evolved things like a loss of dispersal mechanism, loss of seed dormancy, so that hypothetically all the seeds you plant will germinate at the same time instead of waiting until next year. An increased resource allocation to the harvested portion of the plant in many cases. And then also sometimes a really dramatic mating system change, a shift, for example, from outcrossing to selfing, or in some cases really cool shifts, for example, in grape, from being a dioecious species to being a monoecious species. And these are pretty major phenotypic transitions, but we know that most of our domesticated plants are less than about 12,000 years old. It's really interesting, when I started working on plant domestication questions, people kept trying to push the date of domestication earlier and earlier, and now it's kind of gone the other way, and we're realizing that domestication maybe didn't happen as early as we thought, that there was just a lot of farming before domestication. And so the date of rice domestication, for example, has kind of been brought forward in time. We used to think it was older than we currently think it is. Nonetheless, all these changes have occurred within roughly the last 10 to 12,000 years, and often they've occurred much more recently. So we can assume in some cases that strong selection has occurred to affect these changes over a short period of time. One really great example of the effects of selection during domestication is demonstrated by repeated evolutionary events, right? Things that happen again and again. And one of the best examples of that is the loss of dispersal or loss of shattering in many domesticated cereals. So of course in wild plants, you want the fruit to ripen and fall away from the inflorescence so that it can be dispersed. But in domesticated plants, you want the grain to stay attached to the inflorescence so that you can grab a handful of stalks of rice and then harvest them with a sickle, which is what I learned to do when I was at Erie with Susan. And I did not cut myself, so that was also really awesome. But we've seen this trait evolve again and again, non-shattering or loss of dispersal. And just interestingly, from a genetic standpoint, it's cool to think about the genes underlying these repeated evolutionary transitions. For a while, people were looking at the genes underlying shattering, and they found that in most species, it was a unique locus in each domestication event. And that was true until recently in about 2012, when Lin et al. Published this really cool paper about the SH1 gene, which was thought to be important for shattering certainly in sorghum, also potentially maize and rice. So this has led to kind of a breakthrough, and people have been trying to look in more detail about parallel evolution at the genetic level across these domesticated plants. All right, so that's selection. we all know that artificial selection occurs during domestication. Everybody's fine with that. The flip side of that is the random forces that have a major effect during the domestication process, right? And these are usually characterized as genetic bottlenecks that accompany the domestication process, right? So these cause a genome-wide reduction in variation in a domesticated species, and this leads to the fixation or loss of alleles and phenotypes by chance alone. And I really want to emphasize that we tend to think of a bottleneck as I often think of it as a genetic process. It does also have an effect, just as it fixes alleles in the genome, it can also fix phenotypes and can contribute to differentiation between the crop and its wild relatives. So it's not all about selection, it's also about drift or genetic bottlenecks. So most of what I've told you about domesticated plants comes from studies that have been conducted in annual plants. And we tend to focus really on annual grains and annual legumes, and I've shown a couple of them here. And then we throw in sunflower as a token non-grain, non-legume domestication system. And of course, economically and from a practical standpoint, this makes sense. These crops really feed us and they really drive our agricultural economic system. But unfortunately, this has left us with relatively little information about how perennial plants are domesticated and how they respond to artificial selection under domestication. And that's, of course, really unfortunate because perennial plants have been domesticated in every major agricultural center around the world. I'm sure we all have a favorite domesticated perennial that we could reference. And while they tend to not be quite as important as annual crops in terms of monetary value for these different regions, they often have a lot of value just to humans in general, of course. And I was just talking to some people at lunch. We brought up the almond situation in California, and that's a perennial plant that's having a major impact on water usage and economics in that state right now. So we know relatively little about perennial plant domestication, and yet they are really important to us on many levels. And I'm interested in knowing a lot more about how perennial plants are domesticated and what the genetic basis of that domestication process is. And I'm going to be talking about my work dealing with domesticated apple. This is work that I started in my postdoc and that I've continued as I arrived at UM-Duluth in Minnesota. So many of you, I think, are probably quite familiar with apples. You're from a state that's famous for your apples, and of course I have people from Geneva who know a lot about it. But for those of you who have not caught up on all the details, let me give you a little review here. So Malus domestica is the domesticated apple. Its major wild relative, the one we think that contributed the most to its genome, is Malus sieversii. There's also possible genetic contributions from Malus orientalis and Malus sylvestris, which are also diploid wild apples found in Europe and Asia. It's a relatively ancient perennial crop. We don't have a good fix on the date of origin as far as I know, but we do have evidence that it was being clonally propagated by 3,000 years ago, so that's kind of the most recent estimate or the upper limit on its origin. And like many perennial fruit crops, it's outcrossing in the wild, self-incompatible, but it's clonally propagated under domestication. So we graft these scions onto rootstocks, and that's how we propagate the crop. There's a lot of cool things about working in apple. There's a sequenced genome, it's a diploid, it's relatively tractable in many ways. But one of the things that is sometimes overlooked is that there's really good historical records for Malus collections. And for example, one of the oldest trees that I've found data for, the first written record for it occurred in the 1200s, which is really awesome. And another cool example of that is this variety. I pulled this picture from the Grin system. And this is a variety of apple that you can order from Geneva, I think, or it should be available at some point. It's called the Flower of Kent. And the little entry next to it says that hypothetically, this is the tree that Isaac Newton was sitting under when an apple fell down, right? And he started thinking about gravity. So it's just one of the many cool stories about apple. All right, so here's the range of the wild relative of apple, Malus sieversii, has a range through Kazakhstan, Kyrgyzstan, and then also far western China. And the most recent estimates of its domestication indicate that it probably originated here, right on the border, or right on the far western side of China and the surrounding countries. And then it moved westwards to Western Europe, picking up genetic contributions from Malus orientalis, and then finally Malus sieversii, which is found in the farthest western part of Europe over here. Of course, Malus domestica has now moved around the world. It's farmed in temperate regions of the northern and southern hemisphere. You guys grow a lot of apples here. We grow a lot in Minnesota. There's also great apple production systems in South Africa and New Zealand and different countries in South America. All right, so I want to talk about two different elements that deal with understanding the genetic effects of domestication in apple. And I'm going to talk about two different approaches to understanding this. One using neutral markers that I completed as part of my postdoctoral work at the NCGRP in Fort Collins, and another one using a more genetic or gene-based approach. So I'll just start with this one using neutral markers. And to talk about this, I want to go back to this idea of genetic bottlenecks and what we know about them from annual crops. We know, of course, that there's often a genetic bottleneck at domestication where variation is reduced from the wild to the landrace form, and then potentially another reduction in variation with crop improvement as we move from landrace varieties to modern cultivars. I have to say this is the only audience I've ever talked to who understands this step and that I don't have to explain it anymore, so props to you guys. So we know there's genetic bottlenecks, everybody knows this, but as I started to get into the literature, I found that people kind of had these estimates of the severity of genetic bottlenecks that I didn't know if they were real or not. So when I got a chance to work on a paper dealing with domestication with my colleague Allison Miller, I took it as an opportunity to really survey the literature and see the severity of a genetic bottleneck in an annual crop. And these are all figures from different species. Sometimes multiple species or the same species might be in here multiple times in some cases if there's been different studies. But on average, there's a 40% reduction in genetic diversity as you go from a wild system to a domesticated system in annual crops. Obviously, also a huge range across there, some with really severe bottlenecks, some with mild bottlenecks. And this is a problem. Obviously, if you're a plant breeder, low genetic diversity can make crop breeding difficult. If your wild species is still extant, you can go back to it and try to recover some of that variation and useful alleles, but that can be difficult in some crops. And I also want to point out, if you're having a really severe genetic bottleneck, that's bound to affect crop differentiation from wild relatives. If you're losing 80% of genetic variation, you're definitely losing some phenotypic variation and you're fixing some phenotypic variation in that process. One really cool example of these changes that occur unintentionally during domestication is from rice. This is a paper from 2012 where they found that this gene called PISTL1, which regulates phosphorus uptake in low nutrient environments, was completely deleted from most of the genomes of elite Japonica variety rice cultivars, but that it was present in most of the indica rice varieties. So this is an unintentional genetic transition that has accompanied domestication that actually is really bad for the domesticated varieties that we're trying to grow. All right, so that's annual crops. We kind of knew that the genetic bottleneck might be severe in those systems. What about perennials? Well, in perennials, on average, they retain about 90% of the variation that's present in their wild relatives. And there's a lot of reasons for this. Perennial crops tend to have multiple origins. They tend to hybridize with their wild relatives. These are all things, of course, they have much longer generation time. That's a huge transition as well. So we can talk about those reasons more later. But I will point out that one really cool kind of rule of thumb thing is that the most severe bottleneck that I was able to document for perennial crops is about the same as the average bottleneck for annual crops. So it's a really different process of domestication at the genetic level. All right. So we know there's a severe bottleneck at domestication in annual crops. I also talked a little bit about this improvement bottleneck. And again, I went back to the literature to see how severe this transition was. And again, in these comparisons, I'm only comparing the modern cultivars to these landrace varieties. So I'm looking at how much variation is lost during this transition, not going all the way from the wild to the modern varieties. And in this case, I was able to, or the average from the literature showed that you lose about 25% more variation in that step. So you retain on average about 75% of the variation. Again, there's variation, but that's what's in the literature right now as an average. In perennials, we have a really mild domestication bottleneck. Perennial domesticates are retaining a lot of variation. But of course, we'd like to know what happens during crop improvement, or what has happened during modern breeding of these perennial varieties. And so I wanted to look at this using apple because it had such great historical records, and I thought it would be a good chance to take a look at this. So I did that using 2 different data sets, and this was data that was generated partially at Geneva. Actually, this was part of a large collaborative project, and then I analyzed it during my postdoc in Fort Collins. And I had 2 data sets. They're just SSR data sets, microsatellite data sets. One has relatively few markers, but a large number of samples, well over 1,000. And the other one has more markers, but fewer samples. The results are the same from both data sets, so I'm just going to show you the results from the 9-marker data set here. So first, looking at the domestication bottleneck using these neutral markers, I wanted to see whether apple fit the pattern or not. If there was a bottleneck, we would expect to see lower genetic diversity in Domestica. But when I looked at the actual data, I saw that there was no significant difference in genetic diversity across these species. But if you wanted to do a direct comparison, Seversii or Domestica retains about 95% of the variation present in Seversii. So this fits very well with what we know about perennial crops from a review of the literature. Now, that was kind of expected, but let's dig in deeper and look at this improvement bottleneck. And I was really interested in using apples for this because I could split up the data set not by species, but by year in which the apple variety first occurred in the written record. So I used passport data from Grin that had been collected over the years, some scanned copies of notes that from the original donations to the gene bank and things like that, and a lot of old books to get these dates. So I had apples that were described up to and through the 1600s, and then for each following century, the 1900s I divided into 2 chunks, before 1950 and after 1950. And hypothetically, if there has been some improvement, you might see a reduction in genetic variation through time, with the 1950s to present having the lowest level of genetic variation. That's if these fit the same model as annual crops. But as you've probably guessed by now, apple does not fit that model because it's a perennial. So there's no significant difference in genetic diversity across these different centuries. And if you want to do a direct comparison, modern Malus domestica has 99% of the variation present in the in varieties from the 1600s and previously. So extremely low changes in genetic variation through time in domesticated apple, at least based on these markers. And so where does this put us? So in general, perennial crops have mild genetic bottlenecks at domestication, and apple definitely fits that. Apple also has almost no sign of an improvement bottleneck, although apple has been improved. It's not like it's been in stasis. There's just no loss of genetic diversity due to modern breeding techniques. If you go to the literature, on average, and I have to say this is based on not very many studies because not very many people have looked at this, on average, perennial crops actually show a similar improvement bottleneck to annual crops. If you look at the figures that are present, shows that perennial crops lose about 25% of variation in that improvement step. But I really think we need more data about this transition in perennial crops for crops in which that has occurred. So these perennial crops have really mild genetic bottlenecks, but I want to emphasize they still show genetic and phenotypic differentiation from their wild relatives. So if I run a structure analysis on this data, or if I build a network, I can definitely tell the difference between Seversii and Domestica. But despite that, Domestica still has a lot of the variation that's present in Seversii. So this is a really different system than what's present in annual crops. I also want to point out that, and I do this more for general audiences than for you, all of these samples are from the collection at Geneva. That collection is specifically designed to maintain genetic diversity. It's not representative of what we actually grow as a nation for apple crops. So about 50% of US apple production is dominated by just 4 varieties, and about 25% of our production just goes to Red Delicious. This is not really in New York, is my understanding. It's more out west in places like Washington and Oregon. But there you go, the diversity is not really reflected on the landscape at this point for this particular crop. All right, so now I want to take a minute and tell you about a study that I've just kind of gotten the data for it, and I wanted to show it to you guys and get some feedback from you, dealing with a more gene-based approach to this question of understanding variation in apple. And I just want to talk about the most simple scenario of what we expect for selection on a trait and the genetic effects in a domesticated system. So in a perfect system that obeys all the theory and all the rules that we have about domestication, we'd expect to see 2 things if we looked along a chromosome that contained a gene that was a target of selection. First of all, we would expect that the wild species would have overall higher variation than the crop species just due to a genetic bottleneck. That might be really mild if you're working in a perennial crop. And then as you reached a gene that housed an allele that was the target of selection, you would expect to see very low variation at that point because a favored allele would spread across the population and sweep away variation at that point. And this has been shown in several domesticated plants, TGA1 in maize, or the work that came out of Susan's lab dealing with the RC locus or gene in rice. They basically show this pattern beautifully. But the question is, what would we see if we tried to look at patterns like this in domesticated plants? How well do the patterns that we know from annual plants really translate to these perennial crops? And in particular, domestication traits in apple might be a little bit difficult in some senses because we have important directional selections. So we have, in general, somewhat larger fruits in domesticated apple. We also tend to have some changes to a firmer texture rather than a mushy, soft texture, which can be the case for some wild apples. Although there is some overlap in the distribution between Seversii and Domestica for both of these traits. And then we also have diversifying selection. Of course, that's present in every crop, but it may be particularly important in these perennial crops where the fruit tends to show a wide diversity of phenotypes compared to the wild species. So I've just listed a couple things that are important: fruit color and flavor profiles. And of course, this is actually a picture from the Geneva collection showing a lot of variation in domesticated apple. So basically, my question is, what are the patterns of variation for candidate domestication genes in domesticated apple? We really don't know what the answer to this question is right now, and that's what I wanted to find out. And so the approach I took to this was sequence capture by hybridization. One approach could also be whole genome sequencing. I will be honest, I really wanted to not do that in apple because there's a lot of genomic complexity, segmental duplications, and aneuploidy. And so I wasn't interested in assembling these genomes because I didn't think I'd get a good assembly for them. So instead, I chose to pull down sequences of interest, which I'll tell you about a bit more in a second. And to make this a complete study design, I had to have both background genes from across the genome and my candidate genes, right? So for my background genes, I just chose 4 loci from each chromosome and made sure that they were low copy by blasting them against the genome to make sure they weren't part of a large gene family. And then I also had 49 candidate genes pulled from the literature. And these candidate genes deal with traits that we think are important to humans or to apple breeders. Things like taste, softening, fruit abscission. So does the fruit stay attached to the branch or does it fall off before it's ripe? Things like that. So these were chosen, of course, without regard to their genomic location, just based on their function. So that's 117 genes. It's a relatively small number of genes compared to the genes in the genome, but it covers what I'm interested in terms of understanding the dynamics in the system. Many of you may already be familiar with this system, but in case you're not, if you're interested in it, I use the company Microarray. They basically make little RNA baits that tile across your genes. You make an Illumina library, and then the baits that they give you are biotinylated. You mix them with your library, then you put in streptavidin-coated magnetic beads, and they pull down, just hypothetically, just the fragments that you want. You wash the rest of the library away. Very elegant, in theory. Okay, so I got a bunch of samples of apple from Geneva. Thomas Chow found them, and I sent him a list, and he found them in email, mailed them to me physically, extracted the DNA, did the standard Illumina library preparation, and then just did the hybridization to these baits or these probes and pulled down those fragments that I wanted. Then after that, it's a standard Illumina paired end run. And I said that I was doing this. I have a very small lab. This was actually mainly done by my crack undergraduate, Marshall Wedger, who's here at that's a picture of him at his first botany meeting. And now he's a grad student at Washington University. So if you know anybody who wants to come and work on apples, not at Geneva, but in Duluth, have them come and talk to me. I need a replacement for Marshall. All right, so in this project, I started with 96 samples, and only 43 of those survived all the rounds of QC for this project. So library QC, post-hybridization QC, and then QC for just quality of reads and things like that. But I still had a good distribution of samples, 21 Domestica, 15 Seversiae, and then some samples of Orientalis and Sylvestris. All of these gene models were based on the version 1 of the Malus domestica genome. We're now on version 3. There was a lot of activity between when I started the project and when I got the data. But I just aligned those reads separately for each of those gene models that I downloaded from that first version of the genome. And then this is a very gene-based approach. I wanted to just align all the species within for each gene, take those alignments, and then calculate pi for each species and each gene, and then each gene class. So again, whoops. Oh, we are sorry. Sorry, you guys. I've literally never had this happen to me during a presentation before.
Oh, but it's the first time, correct?
Yes. See if I can just get back to that. Anyway, my question is, what is the difference between the candidate genes and the background genes for these different species, right? So we already know from the previous study that at the background genes, you expect variation in Domestica to kind of match the wild species. You don't expect much difference there. But hypothetically, there might be some lower levels of diversity in Malus domestica for these candidate genes versus the other species, because these are candidates that may have been the target of selection during domestication. I should say I only got through the first 20 candidate genes and 17 background genes for the data that I'm going to show you because I just got the data back recently. And this talk motivated me to analyze it, so thank you. And what I found was that levels of pi are higher for these candidate domestication genes, so that's nucleotide diversity, rather than these background genes. Not expected at all. So what's happening here, right? What's going on? I think there's a couple possibilities. One is that if this is real, that there has been diversifying selection for many traits in apples. So instead of seeing a bottleneck at these alleles, I need to put them into different phenotypic classes and see if there's lower diversity in different types of apple for these genes, and I have 49 different genes, so it'll just depend on that gene. The other thing I started to think about as I did this analysis is that I carefully selected my background genes so that there weren't 80 copies of a single locus, but that means that they're not the same as my candidate domestication genes, right? Because some of those are in big families, like the waxy genes. I have like 6 different copies of those. So could just be higher diversity. Very likely my read mapping needs some serious thought and consideration to deal with those multiple copies and make sure that what I'm seeing is actually real diversity. So this is very preliminary, but I'm excited to dig into this a little bit more. This is just data. This is the type of data that I feel is really missing for perennial domesticates, and I'm interested in seeing the rest of it. So overall, in terms of understanding the genetic effects of domestication in apple, we know that the crop retains high diversity based on these SSR markers through domestication and through improvement. And right now, we have this interesting outcome that candidate domestication genes have high diversity compared to background genes, but that might be an artifact of how I chose those genes, so I need to look into that further. I want to finish up by thanking you for coming, thanking Marshall Wedger and Abby Schumann at my lab at UMD, Allison Miller at St. Louis University, Gail Volk at the NCGRP in Fort Collins, and then from the PGRU here in Geneva, Thomas Chow and Gennaro Fazio, who have helped me with this apple project. And I can now take your questions.